nome é Bruno. Para iniciar aqui com a primeira palestra de hoje, eu tenho o prazer de apresentar o Dr. Rickson Mesquita, que é professor do Instituto de Física da Unicamp, head fundador do Laboratório de Óptica Biomédica. O professor Rickson trabalhou no Massachusetts General Hospital, em Boston, nos Estados Unidos, e foi pesquisador associado na University of Pennsylvania, no mesmo país. Ele é membro do Conselho de Educação da Sociedade para Espectroscopia Funcional no Infravermelho Próximo e editor associado na revista Neurophotonics. O professor Rickson hoje ministrará para a gente a palestra intitulada Assessment of Global Systemic Psychology with Near Infrared Light and its Application to Neuroimaging. Professor Rickson, obrigado pela palestra e pela participação no nosso evento. Eu gostaria, então, aqui de passar a palavra para o senhor. Obrigado, ótima palestra para todos.
Obrigado, Bruno. Bom, vou estar mudando para o inglês, mas se houver qualquer problema, ou se vocês preferirem fazer em português, eu acho que vocês podem usar o chat para poder comentar, eu posso fazer a troca aqui, tá bom? So, thank you very much. I'd like to thank the BRAINN Congress Organizing Committee for the invitation to speak today. And I'll just want to talk a little bit about one topic that we've been investigating and we've been thinking a lot recently in our lab, which is related to the global systemic physiology, which is usually treated as a problem in the neuroimaging community. And I guess the question we posed in our lab was, well, can we actually make something that's usually used or treated as a problem as a solution to something, right? So the answer, the kind of short answer is yes. Systemic physiology may be a problem when you try to image the brain, but it can also provide beautiful and very interesting and powerful solutions and provide valuable data, specifically if you are doing this global systemic physiology with near-infrared light, which I'll go through over the talk. Okay, so to start with, I don't know the attendance and where people come from, so I guess we do have some people that are not from Unicamp. And even the people from Unicamp and from the BRAINN Institute, sometimes we don't really appreciate the diversity of the group that we have. So I just wanted to show, this is the map of the Unicamp. It's a top view of the Unicamp. And the Institute of Physics is kind of right in the central of the university. That's where our lab's based on. And this is kind of some pictures that we have in their lab back in the Institute of Physics. This is a photo of my group and some labs, some facilities that we have there. And mostly we are interested in developing and creating solutions, both in hardware and software, but I guess most important, the integration of hardware with software. I guess I learned over the years that even software methodologies development, they are of very kind of little use or powerful if they don't follow or they're not accompanied by hardware integration and hardware solutions. So what we actually try to do best is trying to integrate and create solutions based on mostly on near-infrared light to image the brain. Okay, and this is a photo of our group back in December last year. So, okay, so why and what we are interested in and why near-infrared light for brain imaging. So I guess I don't need to say much about this to this audience, but I guess when we talk about the brain, there's a lot we can say about the brain, right? In completely different scales, completely different temporal scales, but also in completely different spatial scales, right? So we can investigate the brain at the molecular level, at the cellular level, all these microscopic units, all the way up to the brain itself as a system, as an integrated system that can provide useful information that cannot be it does not simply emerge from the molecular level, right? So the brain is well known by being a complex system, meaning that even if we understand all the molecular details and all the cell communication details in the brain, we cannot recover what the brain can do at the macroscopic, at the system level, right? So that's why you need to study the brain at completely different levels, and there's no way you can recover from one level, extrapolate whatever you learn from one level to the other, right? And over the years, I guess we have learned a lot, both from the structure kind of perspective and the structure organization of the brain, and also at the functional level. And I guess what we really learned is that over the years is that, well, we really have a fairly detailed amount of information about how the brain is structured and how the brain functions, at least at the single structure level, right? So how the brain works, even in the molecular level, right? We know the brain cells and how they work. We know all the neurotransmitters and how they are released. And so we do have a fairly good understanding at the single structure, being it even a cell or being even an organ or a cortex, a part of the cortex or something like that. What we really don't know is how all this kind of function or all this structure integrates and collaborates and cooperates with each other to provide an integrated perspective of function, right? So we really know how the cell works, how the neuron works, but we really don't understand how millions or billions of cells work collaboratively or cooperatively to provide basic functions that we do every day, like learning and memory and things like that, any cognitive function, but even primary functions like visual or motor functions, right? So really what we cannot understand yet is how we can go from one very single description of the structure to a very integrated level of function. And that's really what most neuroscience studies have been doing recently, right? And the way people have been trying to investigate that is with some very specific functional activation protocols, right? So most of the times you just put some probes on. It can be from different imaging techniques, and you try to probe the brain and try to probe different regions of the brain over time, right? Sometimes you use a task. Sometimes you just use baseline, right? That's typically called resting state functional connectivity. You can do a lot in this sense, and but most importantly, I guess, whatever technique you are using, there's a very low, the signal to noise ratio of most of the techniques that we use are very low, which forces us to repeat the same protocol over and over again, right? So at least we can try to remove a little bit of instrumental uncertainty if we try to repeat the talk or try to repeat the task, or even when you're doing functional connectivity or resting state, you try to repeat the different resting states protocols to hopefully get rid of some of the uncertainty due to signal to noise ratio, right? And one thing that actually people don't really appreciate is actually the major noise contribution does not come from the technique itself, but it comes mostly from the physiology that we have in the brain and that we are not interested in when we are trying to understand how the cortex works. So really, this is the best, the major part of the noise that we have, which we typically call physiological noise, and that goes independent of the technique, right? And to actually probe the brain, we have very different techniques that we can use. They all have different characteristics, and they work under different spatial domains, under different temporal domains, right? But I guess one thing that we can appreciate, and people have been appreciating that since kind of the very beginning, right? One technique that can actually cover very different scales from both spatial resolution and time resolution is optical imaging, right? So, which is represented in blue in this plot, right? So we can actually use light to probe the brain at very fast resolutions until very long time delays without any contralateral or any collateral effects, right? And the same from the spatial resolution, we can go from very rough spatial resolution to very fine spatial resolution of up to millimeters and micrometers of resolution in the brain. Of course, this actually comes at expense of different things, like spatial and temporal resolutions are often a trade-off in any technique that we have. So this optical imaging kind of concept or framework, it does generate a completely different techniques, right? So we can use near-infrared light to probe the brain at the microscopic level, right? With very different techniques, confocal microscopy to photon microscopy, more recently kind of super resolution microscopy, all the way up to a very coarse resolution, spatial resolution, but it's still very detailed temporal resolution, which is basically infrared spectroscopy, right? Most related to near infrared spectroscopy, usually called as or referred to as NIRS in the field, right? So we can actually go from all the way to the micro level and try to understand the cellular behavior, the way up to the system level and try to understand the whole system behavior. Right? And the reason we can do this is related to the absorption of light by the brain. So just briefly here, when you use near-infrared light, I guess the major chromophores that we have inside the biological tissue is hemoglobin concentration, right? So by shining light at the near-infrared, you are mostly sensitive to oxy and deoxyhemoglobin concentration. If you try to shine light above the near-infrared and go to the infrared portion of the waveform, you are then more sensitive to water content and glucose metabolism. But and if you go below the near-infrared, visible, and also UV light, then you start to be more sensitive to molecules or very specific proteins like the RNA and the DNA of the cells, right? So by shining light at the near-infrared portion, you can actually get good penetration because absorption is quite low, mainly because of the skin absorption is quite low. And at the same time, all the major absorption you have is related to hemoglobin function, right? So that's actually the power of any near-infrared light, independent of the spatial scale you're looking, independent of the technique you're looking, you are very sensitive to blood and basically to oxygen concentration in blood, which can tell you about hemodynamics in any tissue, but specifically in the brain here, right? And you can use this contrast to really understand and to really track brain function. So this was one of the very first experiments we've done over 10 years ago, right? Where we actually tried to correlate images or optical images that we are seeing with near-infrared spectroscopy with the electrophysiology recordings from an electrode inside the brain and the motor cortex, the sensory motor cortex of rats. And then what we can see is actually just doing a very simple whisker barrel stimulation. We can see, well, the electrophysiological recordings both at the kind of long scale, right, and the multi-unit activity at the very fine scale. And as you start the activation, what you see is an increase in blood flow, and then an increase in oxyhemoglobin, a decrease in deoxyhemoglobin. And if you sum oxy and deoxyhemoglobin, you have the total hemoglobin concentration in the tissue, which will be related to blood volume, right? So you typically have an increase in blood volume, which is represented by an increase in total hemoglobin concentration. One thing that I actually try to call attention here is actually, if you're trying to image brain function, you can see that flow goes up a little bit quicker than oxyhemoglobin and deoxyhemoglobin starts to change, right? So really, and I'll talk a little bit more I'll explore a little bit more on this, but since the very beginning, we know that flow can be a better marker of brain function than hemoglobin or oxygenation in general, right? It spikes early, it spikes higher, so you have a more contrast to noise ratio, and you have a fast response to brain activation, which actually is more accurate to the changes in electrophysiology, right? And the reason we see all these changes in hemodynamics, it's because it's coupled with the neuron activity, right? So if you start with neuron activity, that will increase electrophysiological parameters, which will demand for more metabolic contrast. So that will shoot an increase in oxygen consumption, right? Which is typically measured by the cerebral metabolic rate of oxygen consumption, or CMRO2. It also increases glucose metabolism, which is what people typically use in PET to image or to track brain function. And this metabolic demand is met by a hemodynamic increase, both in blood flow, in oxygenation, and blood volume, right? So this is all the framework that we really don't understand the details still, but we do know that this is coupled since the end of the 19th century, right, through neurovascular coupling. So there is a vascular response that is triggered by the neuroactivity, and this is really what is behind most of the neuroimaging techniques, right? Because most of the neuroimaging techniques are actually measuring oxygen concentration, be it bold in fMRI, being NIRS in infrared, in optical imaging. So these are all tracking oxygen concentration in blood, and we try to relate this with neuron activity, right? The point is actually, what are actually the origins of this oxygen signal, or this oxygen level dependent signal, right? And this is something that we take for granted, right? We always assume this, well, there's neurovascular coupling, and whatever we are seeing changes in oxygenation can be related to neuro activity during a functional stimulation, because we are trying to find correlates of whatever task we're doing. So we try to find temporal correlates, and we typically attribute any changes to neuroactivity. But really, if you think about the origins of this oxygen signal, there's much and way much more than this, right? The first thing, we don't even need to go anywhere besides just thinking about the origin of the signal, right? Because if cellular activity triggers brain metabolism, that will trigger oxygen or brain oxygenation, at the same time, the cellular activity can trigger directly blood flow, right? So there's this neurovascular coupling relationship is way more complex than we think, and it doesn't even make sense to talk about neurovascular coupling and neuroactivity, but it makes way more sense to talk about a neurovascular unit where flow and the cells, right, the endothelial cells from blood vessels are actually integrated or perform integrated actions with the neural cells, right? So all those things are related, and one thing that we don't appreciate is that the cellular activity will demand energy, and therefore will demand oxygen. So this path on the left will try to decrease the cerebral oxygenation. On the other hand, the cellular activity will trigger an increase in blood flow, which will actually try to increase brain or cerebral oxygenation. So in the end, whatever we are measuring with BOLT or with NIRS, it's the result of a competitive effect between metabolism trying to decrease oxygen and flow trying to increase oxygen. Most of the times, flow increases more than oxygen decreases oxygen. And that's why we see an increase in oxyhemoglobin and an increase in oxygenation in general. That's correlated with brain function. But that may not always be the case, and I'll show you a little bit. And even when that is the case, this competitive effect is always trickier or more tricky to interpret, compared to a cleaner signal like blood flow or brain metabolism, right? So, and this is just one very simple example that we did a long time ago. So you can actually, it turns out you can actually measure blood flow with light as well through a different technique other than ears called diffuse correlation spectroscopy, or DCS. And so with light, you can always measure both oxygenation with NIRS and blood flow with DCS. And this is one very simple experiment that we performed in the spinal cord in a sheep model. And we were inducing ischemia in the spinal cord. And you see that from the moment you induce ischemia, I don't know if you can see my pointer, but from the moment that you induce ischemia here, and we can see that by the changes, the sudden changes in pressure, both the femoral and the carotid pressure, blood flow goes all the way, blood flow is black, right? So it goes all the way from 0 to minus 100%. So you almost immediately, in the first minute, you constrain blood flow. But because oxygen signal is a kind of competitive fact between the metabolism and flow, what you see is also a decrease in flow because there's no decrease in oxygenation because there's no flow coming in. But there is this competitive effect that actually slows down the oxygenation decrease for over 5-10 minutes, right? So that just is a clean cut show that really whatever you are measuring, if you're tracking oxygenation, you are slower than tracking flow, you are less sensitive than tracking flow, and therefore it's very hard to track whatever you're seeing, right? So I just mentioned, right? So diffuse correlation spectroscopy is one technique that we actually, and that's for this reason and for this experiment that we just mentioned, right? Here, that makes our lab think, well, maybe blood flow is a better marker to track brain function than oxygenation, right? And we did, in fact, we spent a lot of time and effort trying to image the brain with flow and not with oxygenation, right? And for this reason, we developed one technique that it's called diffuse correlation spectroscopy, which can actually measure perfusion and blood flow at the microvasculature, and that will trigger, that will actually measure blood flow, which is definitely a better signal than oxygenation to track brain function. So this is just one picture of the system we developed here in the lab. It was developed by Rodrigo Forte when he was my PhD student. So, and the way it works, I'm not going to go into the details, but basically, if you shine light in any sample when the sample is moving, and if you are able to measure this intensity, the scatter intensity at very fine, very small temporal intervals, you can actually look at the changes in intensity that happens very fast and correlate these changes in intensity with the movement of the cells. And it turns out that in tissue, most of the cells that are scattering light are red blood cells in blood. So you have a good proxy of blood flow by tracking intensity, right? And it does work. So this was the very first demonstration over almost 20 years ago that DCS can track brain function. So this was a finger tapping experiment performed at the University of Pennsylvania by Turgut Duran. And you can actually measure oxy and deoxyhemoglobin, but you can also measure in blue the blood flow with DCS. And you see also a change of good marker. And one interesting part, well, so it does work, right? It was validated and gone far in the past 20 years or the past 18 years with this technique. Another good thing about measuring flow is because if you're trying to measure combining blood flow measurement with DCS, with cerebral oxygenation, you can also measure brain metabolism, right? So, and this is something that it's very, because really, whatever happens in the brain, it will happen because of metabolism. And that's really the whole, what you really want to measure, right? And we've gone a long way through this. I'm not going to talk a little bit much about blood flow, but this is one experiment that we've done in the neuro ICU, where we can actually use these optical techniques to measure flow, metabolism, and oxygenation. And we can see that this was in a patient that was with a brain injury, a subarachnoid hemorrhage, and it was in the patient was at the neurocritical care. So we could actually measure blood flow, oxygenation, and metabolism at the patient's bedside, and we could actually anticipate a vasospasm, right? So, and you can see that the changes in CMRO2 are way higher, so you are actually more sensitive than any changes either in flow or in oxygenation here by the oxygen extraction fraction. So really, trying to measure flow and trying to combine flow with oxygenation is the best of both worlds. Trying to just measure oxygenation, it won't move you very far, mostly in terms of interpretation. So the natural question is why we keep trying to use NIRS, why we keep trying to use BALD to image brain function. The answer is quite simple. We don't have a good signal-to-noise ratio with the flow techniques, right? Be it with the other techniques that we came up to measure flow, they are with very small signal-to-noise ratio, and that's a limitation. Be it DCS in NIRS, DCS in infrared, in optical imaging, has a smaller or a lower signal-to-noise ratio than NIRS. In fMRI literature too, BOLD or ASL, right, which also measures perfusion, has a lower signal-to-noise ratio than BOLD. So there is the good side of measuring flow, which is really you have a cleaner signal, but there's a downside right now, which is related to can we improve the signal-to-noise ratio, the SMR, of these techniques, right? And really, what I do think is that we should be pushing forward in the neuroimaging development community is really trying to get perfusion measurements better instead of just do whatever we can with near-infrared spectroscopy. All right. This is just another example what we can do with by measuring blood flow, but I'm not going to go on this just for sake of time. So the point is blood flow really a measurement and a holy grail for imaging neurovascular coupling? And I guess that's the point. We do have an issue of SNR, which probably it's a matter of development and a matter of ages and decades to improve these techniques. But even if we can get good signal and good signal-to-noise ratio with blood flow techniques, either ASL in MRI or DCS in optical, it still does not tell you everything about neurovascular coupling, right? And the main reason is because there's a lot of neurophysiological mechanisms that are not necessarily related to neurovascular coupling, but they also happen in combination with brain function, right? So, and therefore, whatever technique you're using will be sensitive to this as well, right? So a lot of things can actually factor in when you are trying to measure brain function. You have systemic contributions to the signal, right? Arterial pressure will drive flow, and therefore will also drive oxygenation, right? And arterial pressure can change in very fast scales in the cardiac cycle, but it can also change in a very long scales with other contributions like Meyer waves, right? So depending on the blood gases, they will change the amount of CO2 and the amount of O2 that you have around the vessel. We will also change. CO2 is a vasodilator in the brain, so we'll change blood volume, we'll change blood flow as well. Therefore, we'll change oxygenation. So there's a lot that can go on in any measurement, be it blood flow or oxygenation, be it with MRI or with optical signals. that will factor in addition to the neurovascular coupling that we are trying to measure and to track whatever we are doing neurofunction or function neuroimaging, right? And there's a lot of signaling and even resting state signaling going on that it's not really related to neurovascular coupling, but just to provide a sustain basal activity of the body itself and not only on the brain, right? So there's a lot of subcortical pathways there that comes from down above, right? And we will also change brain signaling, and that's not necessarily related to any activation or to any task or to any stimulation, or even the lack of stimulation thereafter, right? And the bad part is that most of flow control comes from outside of the brain, right? So most of the times, whatever technique we're using, we are interested in looking at the cortex and see what's happening in the cortex, but really, whatever is controlling the brain is outside of the cortex. We typically trash all this signal, right? We trash everything that comes from the pylorus and comes from what we call non-cortical or extracortical contributions, and we try to look at the voxels that are active in the cortex, right, or around the cortex. But really, what is controlling the oxygenation and flow at the cortex is all the vessels and all the vasculature outside of the cortex, right? So there are a lot of problems, and they are all related to systemic physiology per se, but they are all present in any measurement we are doing. And really, to those interested in this topic, I guess there are two very good review papers from Constantino Iadicola group, one from 2017 in Neuron and another one from Nature last year, which I highly recommend to those who are actually interested in this topic, right? So yes, whatever we're doing, be it with flow or oxygenation, BOLD, ASL, NIRS, DCS, there are systemic contributions that you cannot escape, right? And they do affect the signal. So in what ways they affect the signal? And there's a lot of research that we performed, and also a lot of other groups also performed over the past decades, mostly, that can tell you a little bit about this, right? So the first one, which is probably no surprise to anyone, is that systemic factors may be at least one of the main contributors to this negative BOLD response that puzzled a lot of people in the first decade of the century in the fMRI community, right? So this is one very beautiful experiment by Elizabeth Hillman's group in Colombia. And what they did is actually they used mice, and they used very young mice, which the neurovascular development was not completely fully maturated. So whatever response you're seeing would be mostly driven by systemic factors and not neurovascular factors. That was a very nice and very smart idea. And what they were able to show is actually, depending on a task you're doing, and here they were doing, if I'm not mistaken, it was hand paw stimulation, right? You see a blood pressure changes that depends on the amplitude. The higher the amplitude of the stimulation, the higher the blood pressure changes. And the higher the blood pressure changes, the higher the hemodynamic changes. And these are hemodynamic changes exclusively related to systemic changes and not related to neurovascular coupling, right? So, but the answer and the response you see is literally exactly the same that you would see by neurovascular coupling, at least the same time of HRF, hemodynamic response function, that fMRI community likes to use in their general linear models. And we also, in the near infrared field, like to use, right? And the interesting part is actually, depending on the stimulation amplitude, you don't actually increase blood pressure, but you have a decrease in blood pressure. And this decrease in blood pressure also drives a decrease in blood flow, which would give rise to this documented BOLD or negative BOLD response, right? So this is just to show that basically, even in the absence of neurovascular coupling, systemic factors, and in this case in particular, blood pressure can completely drive whatever response you are measuring, either with BOLD or with NIRS, right? Be it positive, be it negative. And they also follow up these mice, and they show that we can see a completely different changes, and they can show exactly the importance of neurovascular coupling, right? So these images in the right are just imaging the same mice or imaging mice at different ages as they age from the top to the bottom. And what you can see is actually when the neurovascular unit is not maturated, is not completely formed, you see a negative response. And as the mice age, the neurovascular unit is being formed, and then you start to see an increase in oxygenation due to an increase in flow. But because it's not completely formed, it just drops, and arterial pressure drives the changes in hemodynamic activity. And only when these mice achieve the adult age, you see the neurovascular coupling sustaining a response that it's kind of the typical response that we typically measure, an increase in volume, an increase in oxygenation, and a decrease in deoxyhemoglobin, right? So you can actually try to understand, and this has a lot of impact mainly for development studies, right? So, and this is kind of a big deal right now, mainly in the optical community, because it's easy enough to probe infants and neonates and toddlers at very kind of small ages, very early ages. But if you're expecting to see a standard response due to neurovascular coupling, you may not find it. And even in adults, right, depending on where you're looking, if you're not looking at the kind of very local and very primary function, depending on the amplitude of the stimulation, you may drive an increase in systemic physiology that will completely affect your signal. Okay? So this is one, it's just one simple way where systemic physiology can be a problem if you're trying to understand brain function due to neurovascular coupling. Okay? Another thing that we can do is actually, if you think about systemic physiology, you have these slow components of systemic physiology, and mainly the slow components of arterial pressure that are typically called Meyer waves. And they do have a significant contribution to the neuroimaging signal, right? So this was also a very interesting study from Miriam Sell in Boston. And what they did is basically they got resting state data. This was NIRS data, not fMRI data. And they simulated HRFs. And they saw that basically whenever you have people with very low intensity Meyer waves due to arterial pressure, you can actually and quite accurately recover HRF that is simulated. But if you have people with high intensity Meyer waves, whatever you are seeing, it completely disrupts your change. So sometimes it completely disrupts your response. So sometimes the lack of response does not necessarily mean that you don't have a significant response. It just means that this patient has a very high systemic contribution that is masking any response. And that's critical if you are trying to do group studies, because you are putting together subjects with very low Meyer waves with subjects with very high Meyer waves. They do not necessarily will see a response, and that will definitely affect whatever group response, whatever p-value you're looking for, right? And so this is something very important, because it's a cofactor that if it's just ignored, it may completely mask your result in both ways. It can decrease your ability to see changes, and it can sometimes increase your ability to see changes, right? And this also happens in the resting state, in functional activities studies. So this is one study that we did a long time ago, when we saw that basically the systemic physiology can increase the connectivity across different regions, right? So if you don't account for the systemic physiology, you are maybe just looking at correlations that are not really related to the brain. They are just spurious correlations induced by systemic physiology, arterial pressure oscillations. They are global and therefore tend to increase the brain, right? And this study came out a few years ago, and that's a very smart idea from Yunji Tong's group. What they did was they recorded resting state functional connectivity studies with both fMRI, and they derived all the famous resting state functional connectivities that are currently reported with both fMRI. All the 12, 13 networks, I don't know how many, but they did a clever idea. They got one near sensor and they measured the hemodynamic changes in the finger, right? So these are completely unrelated to the brain. It's just hemodynamic oscillations measured at the periphery, in this case in the finger, right? And they correlated the changes in hemodynamics they were measuring with the finger with all the changes that they were measuring in these networks. And what they saw basically is that a lot of the famous functional connectivity networks that we found and report, they turn out to be very well correlated with the correlations that are not from the brain in the finger, which basically suggests that most of these networks, they are not really brain networks or brain-derived networks. They are actually basically physiological hemodynamic oscillations from physiological global physiology, right? And this is more critical from the three main networks: the sensory motor system, the auditory system, the visual, which is the primary. The default mode and the frontal parietal has a less of a correlation, but it still has some correlation, which is significantly different from zero, right? So, so really, most of these networks we see, it can be, or at least has a large part that it's just systemic, it's not neural, right? So it still can be used for something, but it's not what we think what they are, right? And they also lead to a lot of data variability. This is just one very simple study that we performed a long time ago in my lab. So we tried to do a very simple task, finger tapping with FMERS, and we were able to see the kind of standard response that we expect, even with very, very few subjects. 5 subjects was enough for us to see this hemodynamic response at the motor cortex due to finger tapping, both in the spatial and the temporal scale. But if you go to one single participant and you try to measure the same participant at at different days, you see completely different results. If you get the same participant to do the same task over 3 different times of the day, you also see completely different responses, right? So this is variability, and this is also related, and we did that. This is also related to the systemic physiology. If you get the same subject in the morning, in the afternoon, in the evening, you have a lot of different contributions of the circadian cycle, which will affect your signal. Sometimes it may increase your sensitive to brain function or to brain function. Sometimes it may decrease your sensitive to mapping brain related to finger tapping, which is kind of a very simple study. So really, all this systemic physiology also induces to a lot of variability, right? So the question is, okay, so we do have systemic variability, and that may have a lot of effects in whatever results we are doing in neuroimaging. So the question is, can we overcome this? Can we actually do something with this, right? Or can we at least eliminate this to really understand and to really interpret brain-based signals from neuroimaging, right? Well, the good answer is that yes, FMERS can measure these physiological contributions without any neuronal contributions during brain function, right? And the way we typically do that with optical signals is just by measuring source, by detecting light at very short source, very close to the source, right? And because if you go to the Fury, I'm not going to go into the details, but basically if you go into the Fury, the close a detector is to a source, to a light source, the shallower it probes the brain. So if you are close enough, you are not getting any signal from the brain. It's just from the extracortical cerebral, extracortical contributions, which is sensitive to systemic physiology, right? So the point is actually, if you are imaging, if you're using infrared light at with short separations, right, with a detector very close to the source, you are just getting extracortical signal, which is basically systemic and the scalp hemodynamics that I mentioned. And if you're doing the long separation, you are getting cortical hemodynamics, but you're also getting all this extracortical physiology. So basically, by doing both at the same time, you can use the information of the short separation to remove the information of the long separation, and therefore just look at the cortical hemodynamics, right? And that can make a lot of difference. So these are just dramatic changes that we can see in kind of activation tasks, right? So this was a cognitive verbal fluence test. When we looked at one region, right, and with the long sources of separation, we don't see any change. But if we actually remove the contribution of systemic physiology, what we see is not only a very nice change, that a very nice activation where we were supposed to see Broca's area, it's a verbal fluence test, but also you do see an increase in oxygenation, a decrease in the oxygenation, which is really nice and really as we would expect due to neurovascular coupling, right? In resting state, this is also the case. So this is one very fresh study that we just recently published in collaboration with a group at Western University in Canada. So we looked at the resting state networks for 10 different subjects, and what we see is actually, if you just get the data without accounting for systemic contributions, you see a lot of variability across the subjects. From the moment that you actually account for the systemic physiology, what you see is an incredible, reproducible, in this case, auditory network without any systemic contribution. So then we can compare and we can just look at 10 completed subjects because they are very reliable and they are very coherent across subjects. So I can make a very nice group comparison now because I don't have a lot of variability, right? And that allows us to actually perform longitudinal monitoring at the single subject level, which I really think that neuroscience should be about. Right? Neuroscience shouldn't be about group studies; should be about individual level at the individual subject. Right. So this is just one example that we also performed very recently, right? Where we actually trying to image functional plasticity with NIRS in a study during rehabilitation of a patient that suffered a traumatic brain injury. And you can see what is called the specialization effect and the learning effect due to plasticity here, right? So before rehabilitation, what we see is actually this brain who had a huge injury in the right hemisphere. All this kind of somatosensory response moved to the left. And after rehabilitation, the brain specializes and incorporated this new function in a very specific region that was not the somatosensory cortex, right? So, so you can actually image this because you don't have a lot of variability because you're removing variability from systemic physiology, right? So this opens up a lot of studies. So this is just a glimpse of what we've been doing. This is one study that we've been doing with learning. So we can actually measure people in natural environments outside the lab because NIRS is a very portable technique. So people can just wear systems and measure them. And if but they will have a lot of systemic contributions, and if you can remove the systemic contributions, you can really image the brain at the single subject level. This is just learning during a cooking class. We've been performing a lot of different studies like that. Very recently, we've been performing studies during driving, trying to understand the brain during driving, trying to understand how the brain communicates with each other, mother and son, or parent and son. So this is a kind of studies that you can do, and you can do in natural environments with near-infrared spectroscopy, but also you can do it in the single subject level because it's very specific. So to finish, okay, so if you remove the systemic contribution, you can go very far, right? You can go to natural environments, you can image at the single subject level. But the point is, what about the things that you're trashing? What about the systemic contributions? Do they have any valuable information or not, right? So most of the people in the field just are targeting removal of the systemic contributions. And what we asked in the past few years is, well, is there anything there that we can actually use? And the answer is also yes, right? So this is one very interesting study that we performed in our lab, which is patients with carotid atherosclerosis or carotid stenosis, right? And we were just looking at resting state functional connectivity in these patients and compared to control subjects, right? So what we see is if you get rid completely of the systemic component and just have the neuroimaging, both with oxy and deoxyhemoglobin, what you see is that you don't see a lot of changes, right? So basically, the networks you see for bilateral patients or unilateral patients with stenosis is the same with control subjects, right? However, if you look at the whole signal that has both the neuro and the systemic component, then you start to see a lot of differences from severe stenosis and control subjects, right? And indeed, what we saw is that most of the differences is not in the neural signal, it's in the systemic signal that people trash, that people don't even look at, right? So this is one, and in this case, it makes a lot of sense because, well, carotid stenosis is a vascular problem, and the patient is okay. So instead of just looking at the neural correlates of this low vascular, of this low perfusion to the brain, maybe we should be looking at the systemic component to the brain. And what we've been seeing is actually this systemic has more insightful information to understand carotid stenosis than just the neural component, right? This is another study that we did, not in the brain, but in the muscle, because if you're looking at the components in the muscle, you can look at the systemic changes. And we've done that most recently with the hemocovid with COVID-19 through this experiments. I'm not going to go into the details first because I mentioned this in the talk last year, right? But basically what we can see, I'll just mention the results, is actually, if you look at the oxygenation, the muscle oxygenation, you see that the muscle oxygenation of COVID patients is different from the controls. And also the way they respond is different from the controls. So you can actually use this to prognostic the severe of COVID patients just by measuring systemic physiology at the muscle level, right? You don't need to go to the brain to understand what's going on in COVID, right? Or to prognostic values in COVID, right? You can go to the brain to do other things and try to understand the effects of COVID in the brain, but not to actually prognostic effects, right? So, and most important is actually, this is well prognostic that it can separate the different types of COVID, the mild COVID from the severe COVID. So you can actually just look at the systemic component because whatever is going on in the lungs is affecting endothelial dysfunction or is causing endothelial dysfunction to the point that it's causing systemic vasculature, right? And just to finish, so what about all that I've talked was basically, well, you can actually look at systemic contributions using near-infrared light, and this is very natural in FNIR's study. But the question is, how about MRI? Is there anything you can do? And the answer is yes, right? You don't need any optical signal that you can actually collect at the extremities can be used, right? And I showed you how Yunji Tong's group used the signal, it's a PPG signal, right? It's an ear signal from the finger to actually account for the systemic contributions to the functional connectivity studies. But there is even a more clever idea, because in that case, what you're doing is just trashing the systemic contributions, right? If you measure systemic contributions from the periphery and then account for this, you can remove the systemic contributions and end up with just the neural component, the really neural component, right? But if you actually get the near signal at different spots in the brain with different PPG sensors, right? You can actually look at the resting state connectivity of the hemodynamic oscillations, right? So, and this can be done, and Yunji Tong's group started doing this, right? like just measuring the peripheral low frequency oscillations. They're not hemodynamic brain functional networks, brain networks. They are just peripheral oscillations. And but the interesting part is actually they are actually sensitive to a lot of diseases, right? So, and this is one proof of study that they did very recently, 2 years ago, with stroke patients. And they were able to show that stroke patients, you can also look at the resting state functional connectivity of the brain, but you don't need to, because just by measuring the systemic components at different aspects, you see that they disrupt these peripheral systemic oscillations, and they induce a delay in the transit time of blood from the kind of upper limbs to the lower limbs and even to the brain, right? So, and this is way easier, and this is way more accurate, and probably whatever decreases that are well reported in the resting state networks in stroke patients with both, they are just a reflect of this decrease in correlation in systemic physiologies, most probably. And this is a clever idea, and we are actually eager to do this, and go in what I've been calling an optography, in similar to an electrocardiogram, right? So electrocardiography. We are talking about an optography now, just putting a lot of sensors at the extremities and try to understand these peripheral oscillations. We've been doing this for COVID patients that have sequelae, and but also a lot of different diseases affect these peripheral oscillations. Diabetes is one that we are very interested to see, and even the carotid stenosis that I showed before, there may be something at the peripheral level. Okay, so I guess that's all that I wanted to talk. I guess the three main things that I want to draw attention is, first, oxygen-dependent levels are tricky to interpret, right? So both NIRS, they are not as easy, and they're not clean-cut, and they are not a direct reflection of neurovascular crime. There's a lot more going on, right? Blood flow can be a better signal, but it still has a lot of consequences from systemic physiology, right? So, and any neuroimaging technique will also struggle with systemic physiology. However, you can account for the systemic physiology but with near infrared light, right? Both with NIRS or even photoplethysmography or PPG in the case of MRI. Those are all, I think, very important signals that people should be tracking and measuring and using if we are interested in understanding the brain, right? So to finish, I just want to acknowledge a lot of people that are behind these results that I showed you. So I can talk a lot about specifically my lab. Most of what I showed you is the result of Rodrigo, Sergio, and Andres, who did PhD in my labs. They're all at different parts of the world now, and a lot of collaborators, both in Brazil, Minas Gerais, here at Unicamp, the clinical hospital where all the carotid stenosis experiments are done, of course, the brain, and a lot of hospitals in Canada and the US. So with that, Wendy, if you want to have more information, you can look at our website, like for BRAINN.com, also our YouTube channel or our social media, it's Lab Unicamp. So with that, I'll end, and I want to thank for attention. I'm happy to answer any questions you may have, either in Portuguese or in English. Thank you.
Professor Rickson, obrigado pela palestra generosa, na verdade, construiu os conceitos, muito ilustrativa, muitos exemplos. Tá? A gente está aguardando por algumas perguntas, mas eu, pessoalmente, aqui, o senhor deve imaginar, tenho algumas para fazer. Tá? Eu achei muito interessante os exemplos. Então, eu tenho até algumas questões a respeito do NIRS portátil e etc. que eu queria fazer. Mas, primeiro, eu queria saber mais sobre o efeito sistêmico medido com o NIRS, né? E os efeitos que isso gera e as correlações que isso gera nas medidas feitas no cérebro. Primeiramente, esses efeitos, vamos supor, naquele exemplo que foi feito no dedo, né, com o PPU. Esse efeito ele é um efeito homogeneamente observado no cérebro todo, ou ele é um efeito que tem uma grande componente espacialmente dependente, assim, ou essa correlação, ela é global mesmo, ou ela é espacialmente diferenciável?
Excelente pergunta, né? É algo que eu passei batido, mas o artigo detalha bastante. Essa correlação, bom, ela vem, acho que não há dúvida nenhuma que ela vem do dedo, portanto, ela não tem nenhuma componente neural ali, né? Então, é uma componente de fato sistêmica. E como toda assim, se é sistêmica, ela deveria ser global. E de fato ela é global. Tem um outro artigo que eles fizeram antes desse, onde eles de fato mostram que essa componente ela é global. Só que para dificultar toda a situação, né, particularmente de análise de dados, ela é global, mas ela não é simultânea, né? Então o tempo de trânsito para você chegar em diferentes regiões ele muda. Então isso significa que do ponto de vista de análise você fazer técnicas de separação de componentes, por exemplo, não vai ser muito, pelo menos as tradicionais, né? Se você tentar fazer um PCA, se você tentar fazer um ICA, não vai funcionar, porque o PCA parte do pressuposto que essa componente é global naquele instante de tempo, né? Então, isso, do ponto de vista de análise, complica mais ainda, mas do ponto de vista de efeito, é sim uma componente global, e ela está presente no cérebro inteiro. A questão é que ela está presente no cérebro inteiro em diferentes instantes de tempo, né? Então, essa componente pode variar aí entre 1 até mais ou menos 3, 4 segundos em relação ao tempo que é medido no dedo, né? Só o tempo, na verdade, de trânsito do dedo até o cérebro, você já tem aí um certo tempo de trânsito, né? Então essas coisas, elas têm alguma correlação. E aí, se você reparar, não sei se eu consigo voltar aqui rapidamente, mas se você reparar, o que eles colocam, na verdade, é o que eles medem a cross-correlation, né, que é uma correlação cruzada, maximizada para o tempo de lag que você tem a melhor correlação, né? Isso é uma coisa que a gente faz muito também, né? Aquilo que você mede na periferia, no nosso caso, quando a gente está medindo direto a sistêmica, né, na cabeça, aí o tempo de trânsito é praticamente zero, você está medindo no mesmo local, só um pouco acima, né? Mas quando está medindo na periferia, particularmente no dedo, você vai precisar levar em consideração o tempo de trânsito e os diferentes tempos de trânsito no cérebro. Então, de alguma forma, você vai precisar considerar essa situação, né? Existem algumas técnicas que ainda conseguem fazer isso até nas análises, né? Canonical correlation, talvez, o temporal embedded canonical correlation, é uma técnica um pouco mais fina do que PCA e que consegue levar em consideração todos esses diferentes tempos como regressor. Mas sim, de forma objetiva, é uma técnica, é uma medida global, só que vai estar presente em diferentes regiões do cérebro em diferentes instantes de tempo. Então você precisa considerar também esse tempo de trânsito na vasculatura.
Ela é global, mas tem uma componente espacialmente dependente, então. E será que existe uma forma, isso é padronizável? Existe uma forma de mapear essas regiões para esses delays? Existe uma forma de você particionar, será, o cérebro para esses delays? Não sei se eu estou conseguindo ser claro, assim. É previsível, será, saber se as regiões mais profundas, mais corticais, a propagação desse delay, será que ele é constante entre as pessoas? Porque, assim, de fato, quando a gente faz a análise de conectividade funcional, a gente tenta regredir para qualquer efeito global através de artifícios que a gente tem com a própria imagem, né? Então, a gente espera que componentes que são globais, que são distribuídas espacialmente pelo cérebro, sejam removidas, e isso é feito. Tem até discussões da melhor forma de como isso é feito, através do sinal global de Liquor, de white matter, para tentar pegar para pulsação, etc. Mas, no final das contas, o que a gente obtém é um sinal global, né? E o efeito da pulsação, a gente imagina que ele seja uma coisa progressiva, uma coisa que se propaga mesmo, né? Então, a ideia de que exista esses shifts temporais é muito interessante. E por ignorância minha, uma pergunta sincera, você sabe se existe, se é proposto alguma maneira de se corrigir isso, talvez com derivadas temporais, alguma forma de você introduzir uma variabilidade temporal nos sinais globais, uma alternativa, ou se, voltando ao início da minha questão, se existe uma possibilidade de se mapear regiões, de se mapear esses sítios que devem ser considerados como, em termos de delay, de um sinal fisiológico obtido como um eletrocardio, por exemplo, ou de um PPU no dedo, se existe algum esforço para se modelar essa propagação no córtex e se modelar esse shift temporal, especificamente, assim.
Bom, vamos lá, várias perguntas, né? E várias boas perguntas.
Estou
Né?
pensando.
Várias boas perguntas, vamos lá. Bom, primeiro, começando pelo início, né? Parcelar pode ser uma tentativa, mas eu não sei se ela seria muito bem sucedida. Nós nunca tentamos fazer isso, né? O que a gente tenta fazer, no final das contas, é o que você está chamando de parcelamento, né? Em situações de distúrbio cerebral. Então, aí, de fato, assim, o que eu posso te falar é que ela não é igual, né? Mas é esperado que ela não fosse igual, justamente porque a gente está estudando algum distúrbio que está afetando a circulação e o tempo de trânsito do sangue nas diferentes regiões, né? Se você pegar um grupo de conjunto, e aí, assim, se você pegar pessoas com diferentes idades, isso também vai ter um efeito, né? Então, se você pegar um grupo relativamente homogêneo, saudável, eu não sei te dizer até onde esse tempo de trânsito e essa circulação ela vai ser homogênea e, portanto, você poderia assumir uma mesma circulação para diferentes voluntários, né?
Modelo geral, né?
Isso. Agora, o ponto central é que se você está coletando isso de forma simultânea, talvez você não precise de fazer isso, porque você tem esse dado hemodinâmico simultâneo. Não há nenhum motivo para acreditar que esse dado hemodinâmico puramente sistêmico, né, que não tem a ver neural, ele não seria o mesmo, né, em diferentes partes do corpo. Então, se você tem esse se você tem esse sinal o tempo inteiro, num certo intervalo de tempo, aí você tem, talvez a questão é como você vai usar esse sinal para fazer a regressão, né? De fato, assim, regressão de sinal global é uma coisa que se discute desde o início, né? De conectividade funcional, em Bold, essa discussão se propagou para a comunidade de NIRS também, né? Mas o ponto central é que, e todo mundo tenta fazer o melhor que se pode, né? Mas o ponto central é que tudo que a gente tenta fazer são regressões estáticas, né? Então,
geral,
enfim,
sim.
O grande problema, no final das contas, é que você está considerando as correlações, né, daquele sinal naquele instante de tempo com todas as regiões cerebrais, né? Então, essa é um grande problema, né? O segundo grande problema dessa história é que é um problema matemático, que até onde eu saiba não tem solução. Qualquer modelo de regressão linear, como o que a gente costuma fazer para fazer essas regressões, né, ele é um modelo que não sabe lidar com colinearidade, né? Então, aí sendo um pouco mais técnico, né, do ponto de vista de tratamento de dados, né, regressão nada mais é do que a inversão de uma matriz. Só que se você tem matrizes com duas colunas que são muito similares, ou linearmente dependentes entre si, essa inversão vai para o buraco, né? E a gente faz isso ignorando essa questão. Então, a fisiologia sistêmica te traz exatamente esse problema, né? Seja ela vinda do líquor, seja ela vinda de um canal curto, no caso de NIRS, né? Porque a fisiologia sistêmica vai fazer com que essas coisas tenham algum grau de linearidade. E aí pode ter o efeito oposto. Quanto mais regressores você colocar no seu sinal, maior a chance de você estar adicionando colunas colineares, né? E aí você está fazendo essa matriz virar ill-posed, né? E aí essa inversão vai ter um efeito oposto, né? Porque você não vai estar conseguindo regredir aquilo que você queria justamente por um problema matemático. Você não consegue inverter matriz que tem duas colunas colineares. Então, aí é um problema do ponto de vista técnico-matemático. E aí, finalmente, para responder a tua última pergunta, né? Então, como eu poderia usar isso e usar particularmente os diferentes instantes de tempo, né? Uma das formas que você pode, talvez a forma mais... há pessoas, né, com expertise em matemática para fazer isso, certamente não sou eu, para que têm estudado e formas de poder resolver esse problema. A solução mais elegante ou mais eficiente que eu já tenha visto até o momento é um negócio chamado Temporal Correlated Canonical, Temporal Embedded Canonical Correlation, né? Que, em grosso modo, o que você está fazendo é um modelo de regressão linear, só que você vai colocando atrasos do teu sinal como regressores, né? Então, de certa forma, você está usando o mesmo sinal, só que várias vezes com diferentes atrasos, né? Isso é uma das coisas que você pode fazer, mas, de novo, não é tão simples quanto parece, ou algumas pessoas até têm tentado fazer isso de forma simples, né? Mas não é tão simples quanto parece, porque se você dá um atraso relativamente curto, essas duas colunas serão, de novo, colineares, e, portanto, você vai ter um problema aí, né? Então, o que a gente tem tentado fazer no nosso laboratório é fazer todo um estudo, e de certa forma foi onde tudo isso começou, fazer um estudo da fisiologia sistêmica antes de pensar no estudo da fisiologia neural. Então, a gente olha para a componente sistêmica, a gente olha para esses tempos todos de atraso, a gente consegue entender como são esses tempos de atraso do indivíduo a indivíduo, né? E aí a gente consegue usar regressores apropriados, né, que são linearmente independentes entre si, para poder fazer regressões considerando os diferentes tempos de trânsito que a gente enxerga. Então, é mais ou menos isso que a gente tem ido, né? E a gente tem feito isso indivíduo a indivíduo porque o nosso maior interesse hoje eu acho que a gente tem um ponto, tanto do ponto de vista de hardware, de tecnologia, quanto de software e de análise, né, de entendimento do sinal, que nos permite fazer medidas, né, fora do laboratório, e aí o interesse é indivíduo a indivíduo, né? Eu quero saber o aprendizado daquele indivíduo, não de um grupo de indivíduos. Então, por isso a gente tem tentado particularizar toda essa situação de indivíduo a indivíduo. Então, assim, existem soluções, elas estão sendo construídas, né, nesse momento, quando a gente fala, mas elas ainda precisam ser testadas e precisam ser analisadas, né?
Bom, a gente está chegando no limite do nosso tempo. Tem uma pergunta da audiência. Rapidamente eu vou fazê-la, mas eu já fui alertado aqui que a gente está no limite, mas eu vou fazer. É a pergunta do Ítalo César. Ele está perguntando se there is any possibility to correlate NIRS to a structural deformation. For example, the level of cortical dysplasia or epilepsy.
There might be, né? Eu acho que você pode fazer essas correlações, né? Nós nunca fizemos, há outros grupos que já fizeram, que eu tenho conhecimento que já fizeram. Então, assim, a princípio é possível, né? Particularmente com epilepsia, isso é um negócio que é bastante discutido, né? A única coisa que eu acho que a gente precisa tomar um pouco de cuidado particularmente quando a gente faz correlações, né, associadas aí, particularmente usando diferentes técnicas, é que elas estão longe de serem simples, né? E aí isso, na verdade, virou até uma discussão recente, até onde eu tenho acompanhado, dentro da própria área de ressonância, né, com os Brain-Wide Association Studies, né, que tentam juntar fenótipos, né, com resultados funcionais, a validade desses estudos, né? Porque para você de fato chegar a alguma coisa que seja... Qual que é o grande problema, né? Quando você está tudo dentro de uma única técnica, você tem um erro que é sistemático ali. Então você pode até estar errando os valores ou a interpretação, mas se o erro é o mesmo, né, a fonte de erro é a mesma, isso não vai afetar essas correlações. A partir do momento que você tenta usar técnicas independentes, você está considerando técnicas independentes com erros sistemáticos diferentes, que muitas vezes as pessoas ignoram ou particularmente não estimam, né? Então, nesse caso, você precisaria particularmente, né, de algum EEG ou alguma técnica estrutural para poder detectar isso aí, e mesmo quando você está olhando para técnicas estruturais, isso é feito por um algoritmo de computador que também tem um erro, né, que geralmente as pessoas não estimam, mas ele existe. E aí você tenta correlacionar isso com alguma outra técnica, né, que vai ter um outro erro. Então, no final das contas, a correlação entre essas duas coisas de forma independente, ela exige um grau de cofatores muito grande. A única forma de você fazer isso com de alguma forma bem sucedida, né, ou bem confiável, ou você vai coletar milhares, e é literalmente milhares, a gente não está falando mais de centenas, é milhares de pessoas, dependendo da variabilidade dessas técnicas, ou o que você vai ter que fazer é coletar do mesmo indivíduo milhares de pontos. Né? Então, para responder a pergunta, né, é possível ser feito sim, nós nunca fizemos, outros grupos já fizeram. No entanto, é preciso tomar muito cuidado ao fazer esse tipo de coisa, porque quando você vai pegar resultados, né, que provém ou que vêm de locais ou de técnicas diferentes, a chance de você gerar um acaso devido aos diferentes erros sistemáticos é muito maior do que simplesmente usar uma única técnica. Né?
Ok, eu agradeço novamente, professor, pela palestra, pelas respostas, pelas perguntas. Pessoal, a gente vai direto agora para a sessão de pôsteres, tá bom? Então, eu solicito que vocês permaneçam na sessão de pôsteres. Professor, obrigado.
Eu agradeço.